Yeah, I think I pitched this lecture like a week ago, right?
Yeah.
I was like, oh, we'll be in town. He was like, well, we need weeks. And I said, no, I'm going to be here next Wednesday. So thank you for coming out on such short notice. And thank you for, this area has been so welcoming. Anywhere, like from the Goodwill to the Habitat stores, everybody's just so friendly. Like, where are you from? Where's your accent from, boy? You know, like that. It's like, I don't know. So I just wanted to share some slides and tonight. And like I said, I was really active with the South Carolina group, and I'd be honored to, with the little time that we're here back and forth from South Carolina, Jamaica, and Costa Rica, kind of doing a loop. Whenever we touch ground, as much as we can, we will definitely want to not only share what we know, but also learn, because as you know, every bioregion you go to is different. And when you come to South Carolina, I will tell you every single fungus on the floor, on the table, and when I go to Telluride, and I just walk right by the table, you know, really quick, you know, because people are like, what's that, Chad? I was like, I have no idea what that is. I thought you were, never mind, you know. So it's really bioregional specific. So I had this slide. This is one of my keynotes that I kind of spliced together. It's one of my favorites because it's mushrooms, mold, and mycorrhizae. It's all the cool things that fungi do. And you've probably seen splashes of this, either my lectures or other people's research. And I just kind of brings everything together. And I really appreciate it. This is the first lecture of the year. What a good one to kind of grease it in, right? And as I'm ready to get moving. And so if we gaze deep into nature, we'll find the solutions for everything. And I just turned 50 years old in January. Thank you. I don't know if that's a clap for like, he's almost done, he almost made it. Or he made it that far. I don't know which one it is. But you got this. But when I wake up, things changed. I started growing mushrooms. I was introduced to mushrooms, full disclosure, when I was 18 by my drummer. Okay, you start to get the image here that's going on. And but that session that I did when I took those mushrooms, I was like, everything changed. I mean, if you've seen my talks, I was from a military family. I had a scholarship to the Air Force Academy right here when I was 18, and I didn't turn in my application. And my dad went frickin' berserk. And I didn't go into the military. I went in, I joined a band, and I started growing mushrooms instead. All right. It took a while for the Cotter camp, my family, to accept what I was. And I finally started to build a business, and then wrote a book. And now I went back and I lectured at my dad's alma mater, right? Military College of South Carolina. How about that, buddy? Yeah, full circle. And now he's one of our biggest fans. But I wake up like this, and I also see that the mushrooms are first responders. And whenever you see an ambulance go by and you're thinking, where is that going? Or wonder if somebody's hurt, and the earth is hurting. Just like I spoke to the gal that works here, I said, look at this pond. This pond probably has some inputs in it. Right when I got here, I'm thinking about it. I want to fix this pond. And I just rolled into town because I know there's inputs coming in. There's biological and chemicals going into environments. It happens every single day. This has been happening for millions of years. This planet has been bombarded, but the last 100, 200 years, it's been human activity, right? So over 400 million years, we see that mushrooms produce heat, carbon dioxide, and sweat. We all know this is a club. Does everybody know that? That mushrooms produce heat and carbon dioxide, and they sweat. So it's a combustion process, as you can see by these hot tips, and they are out. Mycelium swims through the environment. Not many people think about that they bead a little bit of moisture over their mycelium, and they drill their way through the landscape, and even through wood, like dry wood. That's how they're able to crawl across really dry environments. So that's what I think about sometimes when I go to sleep. Is actually swimming through my own sweat. Everybody do this. Go ahead. Don't be shy. It's the first meeting, all right? Are we getting a video? I can't believe they're doing this. All right, let's go back to South Carolina. I did it. No, and you think about that, and you just I guess it's always the brushstroke swimming through, or maybe you're doing this. But, and then they're beating moisture. And that's what I'm going to be talking about, is a lot of these metabolites that come off of mushrooms. That's what I study, are absolutely amazing, right? So, and in lichens, every time you encounter lichens, over 60% of the earth is covered by lichens. We know nothing about lichens. And these are lichenized fungi and algae together, and they cover rocks. And it's a composite organism. We are composite organisms. I can't remove all of the bacteria and everything out of my body and still be trad. I've tried. And you see these beautiful ascomycetes, these little cup fungus, and you notice these. If you don't have a little lens, you carry around, a little lanyard, get one this year. And if you're not finding mushrooms and it's really dry, start looking at lichens. Those are fungi. They're there. And they're on the rocks, and they're breaking apart the rocks and making soil for plants and everything. So they're very cool. I wish I could find one of these. These are Prototaxides, over 20 feet tall, millions of years ago. And I put this together because Paul's shown some images of Prototaxides before, but I've never seen anyone put the comparison of what the tallest plant on the earth was at the time. 3 feet compared to 20 feet. The tallest plant was a little plant called Cooksonia, and the fungi were towering 20 feet or 17 feet above the tallest plant on earth, right? And that's what something looks like. We found one of these right above our new house in Woodland Park. Come on, guys, wake up. And then now I get to go out mushroom hunting in a new environment. And this is how you immerse yourself. And you learn from a new climate, a new bioregion. And this is me out looking for morels in the spring. You have morels here? Sometimes, yeah. I do the morel lecture every year for Clemson University. I'm like Groundhog Day for morels. I do the temperature data, and I look at everything, and I can predict exactly the 2 to 3 weeks when they hit in South Carolina. And they started to fruit this year, February 25th, right? Because it was a warm winter, and 2 to 3 weeks in that given area, they are gone. So now us going back and visiting, no chance that morels are in that spot. You have to go to a higher elevation, right? So I have an entire lecture on morels. And then those little fungi that you see, all looking all innocent, may not be so innocent at night, right? Tiny little fungi that bioluminesce. There's over 160 different species that bioluminesce on their own. This is the same enzyme reaction that fireflies use, right? And benthic fish and bacteria. They clip off ATP cells and they start glowing at night. Now, why would they do that? Why would a mushroom glow? Right? I found this one on the edge of a tomato field down in Savannah, Georgia. It's a coastal species. You will not see it here. All right? You won't see it here. And this looks like it's about 50 pounds. And I found this at a garden center. This is a concrete mushroom. No, I'm actually going to flip that joke on you because this is a real mushroom. This is Mycorrhizae titans. This is a real fungus. And that is a 5-gallon bucket. That's not a little Barbie prop, right? This one was over 24 inches in diameter. It was huge. And you could actually sit on it. Imagine finding a mushroom and sitting on it, taking a break, right? And this one had a stalk that was 8 to 10 inches in diameter, and it was really thick, right? It was it was super ancient looking. It had a really thick rind that had to cut with a serrated bread knife, and on the inside was all cottony. So they had the architecture of being able to hold your weight. And so I asked the teacher who found this to send it to me. So I've only seen that in South Carolina once. And anybody do tissue culture here, like lab work? Right, so we could clone this, right? It'd be pretty easy. So I needed a piece. So she sent the whole thing. It weighed 50 pounds, right? And she UPS'd it, but she shipped. I said, put some ice packs in there to keep it cool because it was the summer. It's a summer mushroom. She put dry ice in there. So dry ice killed it, and then it froze solid. And so the day that this thing showed up, a UPS guy comes walking in with a box this big at an angle, and the corner is dripping brown juice. And it was like bloop, bloop, all over the floor, and he sets the box down like this. He sets it down, and he just like looks at it, and he's like slowly backs away. And I said, do I need to sign for it? And he goes, no. He goes, I just want to know what the hell's in here. So he waited, and I unopened it up. It was all mush, except for the very bottom of the stem was intact. It was in a corner, and we only needed a little tiny piece to clone it. So we actually got to clone it. Right? So this mushroom is alive and well in the lab, and it's huge. Like I said, it can hold your weight. So I say we plant these out here next year in like a little circle, like a fairy ring, around a fire pit, and then we can grow our own fire pit stools. Right? That's about a 2-gram idea. I can say that in this state. I don't give a shit. So, South Carolina probably get 10 years just for saying that. Mushrooms produce spores. We know that. They produce mycelium. They produce mushrooms again. I grew a lot of mushrooms. If you saw my name attached to Mushroom Mountain, I started that company in 1996, and I just sold it a year and a half ago. All right. I did good with that company. It's a great company, but we have bigger and better things going on now, so I had to release it. So this is pink oyster mushrooms, golden. I'm still growing these in my house. Is anybody growing oyster mushrooms in their house? It's super easy, right? If you're paying for them, they're really easy to grow. I grow them on paper and cardboard in the coffee grounds. Have a whole recycling system in our house. You've got a glass container box, right? You may have aluminum. Do y'all have a mushroom recycling container? Right? We should. Every town should. Metabolites. This is what I study here. Two mushrooms colliding on a petri plate. Interesting, right? Have you ever seen two mushrooms on the same log? Of course you have. Two different species. This is what you would see if the bark was removed. If you could see inside the wood, you would see that this mushroom and these do not like each other, and they are sweating metabolites like these little biological weapons, and they're trying to kill each other. Oh, I thought mushrooms were all rainbows and Care Bears, right? Now they don't like each other. So mushrooms can be predatory, they can be submissive. These are all like people you know, right? Opportunistic, territorial. They can also change. They can be one and switch to the other. It depends on their situation. So this is mushroom behavior. I wrote the book here on organic mushroom farming, and if you don't have it, it's not a bad text. I could see some people might have it. But they took 10 chapters out of this book to make it smaller. And one of the chapters was mushroom psychology, right? What is a mushroom thinking? And they were thinking, what were you thinking by writing that chapter? They were saying, it's just a little weird. So these are some slides that just kind of opened up a whole new window. And if you're into tissue culture, this should. This happened several years ago when I went back to school for microbiology. I was off campus, had my own lab, and I was growing a mushroom culture and pure culture, and every now and then you get contamination right on a petri plate. You can either cut it out, that's pretty risky to open a plate because it just spreads, or you can just throw the plate away and start a new one. So I threw the plate away, and I left it in the garbage in a Ziploc bag. I went off to work for a week. I came back, and I was emptying the garbage, and then I saw this fungus attacking another mold on a petri plate. So I saw what happened was behavior, active fungal, fungi on fungi, parasitism, behavior. And I'm going to tell you what this fungus is in a minute. So typically this fungus is a beautiful symmetrical circle on a petri plate. And then it got out of its arc like this. You see the lines? So it stopped growing and then surged like a little army of cells towards the competitor. This was awesome. I don't have cable, right? I'm just saying, I was watching this when everyone else was watching like Super Bowl Sunday and meaningless shit. I'm watching my Patriot plates, right? And even posting to the 2 Twitter followers I had. I wonder who's going to win at halftime. And then I noticed these little droplets. If you zoom in, I started noting something unusual. Because in a pure culture, typically you don't see metabolites. And why would they defend themselves in a sterile environment? So the minute that this fungus was threatened by another organism, it started to produce its own defense system. That was interesting. Right? So these plates that I pulled out of the garbage changed a lot of things for me about how I see fungal ecology and what we can do with them. Now, when you see, this is frost bolete from the Carolinas. This beautiful, these are natural metabolites that form underneath this bolete. They look like little drops of golden dew underneath. They're chemoattractants, so they attract insects as well as things like mosquitoes and things to pollinate their spores. They also have antibiotic properties in them, which I'll touch on. That's what I'm interested in. Mushrooms and those enzymes, that sweat that they produce. Remember, we all were swimming through our own sweat tonight, right? And they are producing chemical keys, enzymes. Remember, they breathe in oxygen, so they let off CO2. So that oxygen is a combustion process, like your fire. You light a fire, oxygen, CO2, heat. It's a combustion process. When they have molecules that are around them that if they start to get hungry and they need a food source, fungi can switch and turn on man-made molecules. What's the difference? Because lignin in wood is this huge molecule that most things in nature cannot break down except fungi, because fungi have the skeleton key. They have a molecule, or they have enzymes that are non-specific. So they can go in there and say, hold on a minute, let's bust apart this pesticide and let's take what we need from here. Let's take some of those molecules. This was something I did with motor oil years ago. And then I switched to atrazine when I was working for the EPA for my fellowship. They said atrazine was on the red list. So one night I flooded atrazine into the agar. There's no nutritive source in this gel, by the way. I didn't put anything. It's just agar and atrazine. And I took one of my favorite oyster mushroom strains. We all have a favorite, right? We name them, right? Like Stumpy. And then we stuck them on there, and then it ate up the one plate, the labeled rate. Then it ate up double the labeled rate, what the bottle said, the pesticide bottle said. And then I switched to 3 times the labeled rate. Now, if it was working at the labeled rate, that seems like good enough, right? So why would I test it on 3 times the labeled rate?
Remediation.
For remediation, right? How many homeowners follow the directions? When they say you want something dead, they double the labeled rate or triple the labeled rate. So a lot of, I would say, pollution isn't the large company down the street. It's the individual community homeowners collectively contributing to environmental pollution. That's a fact, right? So I did this, and it sat there, look at this, for 10 days. And it didn't do anything. It just kind of stopped, and now I'm watching it like my pet. I'm like, I'm sorry, I fed you this toxic stuff. It's not going to grow anymore. And then I went back and I figured out the concentration, and I was about to write this up. Stuck it in an incubator. Came back, look at this, from day 10 to day 12.
Oh.
Just like that. 2 days. So it grew out, was testing the waters, pausing, and then it shifted its genetics and then started to express an enzymatic solution that it could use to break down atrazine. Just like that. That's fascinating. So then I started to pour, this is in my book, different gels of different concentrations of parts per billion, parts per million. And I used the coloring just for the book so you could see the colors. It looks good too, it looks pretty. then I started to play with after the BP oil spill in the Gulf, I started to figure out how do you get oil out of water and then, treat it with fungi. Because you can't just pull, salt-enriched bioabsorbents out of the Gulf of Mexico and add oyster mushrooms. It doesn't work, Paul tried it, I tried it, and then we developed a system where you could just take an oil slick, and we can do this with a demo. If you have a mushroom fair, it's very easy. You pour oil into, even cooking oil, and then you take shredded wheat straw. You've seen straw at Tractor Supply or shredded, and just break it up and float it, and it sticks to the straw, just like that. And then you scoop it out. The water's clean. What eats straw? The mushrooms do. So now you've removed it out of the environment. It was so easy. So after the BP oil spill, I put out this ad to the whole Gulf. We sent it down to all the extension offices along the Gulf, right? And this was the key words. Anyone can help with the Gulf oil spill. Anyone. You just find a mushroom on the beach. Find a mushroom on a piece of driftwood and turn it in. So anyone who can culture it, right? And you can clone a mushroom that's been dried for months, up to a year. So if you air dry this, find it and air dry it and then ship it, I can clone it 2 months, 3 months, 6 months later, it's still alive. A dried mushroom, dried properly, will stay alive for months, maybe up to a year. So then we got some hits. How many of you ever been down to the beach looking for mushrooms? You have. Yeah, there's not that many people who know mushrooms are growing right on the beach and sometimes out of the wet sand. You're looking for shells. Maybe you should start doing some mushroom hunting along the beach because they're there. They're way more common than you think. Now in that sweat, we've got mushrooms that produce chemical keys. We got antibiotics. Now we have octanol. If you ever smell a mushroom and it smells a little sweet, especially golden oyster mushrooms are very high in octanol. Mangoes are very high in octanol. The yeast that forms on mangoes makes it extremely sweet. And that also attracts different organisms. Now, how can you use this to your advantage? Especially gardeners. So gardeners who use spent oyster mushroom compost. I did see some farms around here that are selling their old blocks. You could buy their old blocks or grow your own mushrooms and put those blocks into your gardening soil, and then it vectors. It becomes a playground for worms because it produces octanol, and that's what worms are looking for, right? They're looking for that chemical chemoattractant because this is pre-digested material. The worms get in there, they eat it, and what goes in a worm comes out. So what comes out of a worm? Castings. It's beautiful, right? Then you have this a couple weeks later, and then you have beautiful castings for your garden. So you have fertilizer. What is also happening in that sequence? Let me back up. What if this straw or what if your soil had pesticides in it? The oyster mushrooms are sweating and breaking down those precursors to those pesticides. The worm is also a bioreactor inside. It has specialized bacteria that can break down that pesticide, those biotransformational molecules even more, even better. So you have mushrooms bringing wine to the party, right? And the worms are waking up on the couch. Native orchids. I'm not sure what orchids or what plants are growing here, but I do know in South Carolina in the Appalachian Mountains it's very plant diverse and rich over there. All of the native ground orchids in North America, fungi are responsible for opening up their seeds, right? They cannot germinate by themselves. So they have a little fungus called Rhizoctonia. You can see these pine trees, these conifers in the back. The fungus kills the pine tree, right? And then the fungus lives in the ground. The orchid seeds are underground. The fungus pries open the orchid seed like a little oyster knife. It's a little spring. Boing, and it opens it up, and it lets the seed out, right? The plant germinates, and then it lives with the plant underground. This little orchid, the fungus and the orchid are their best friends. They're best roommates. And as soon as the plant, 5 to 7 years later, comes up and starts to photosynthesize, the plant secretes a toxin, and then it kills its roommate. It kills the fungus. Right. You may ever have a roommate like that? You know, roommates, yeah, you have this is your shelf in the fridge, this is my shelf. This is way beyond that. I think just the fungal biology is amazing. Right? So the next time you're out and you really appreciate mycorrhizal fungi, right? This collaboration of this increased surface area of the plant for water absorption. And now you know that they can defend themselves against certain bacteria and certain fungi. So having a fungal filament like a wetsuit on your body in the soil is a benefit, a huge benefit, right? It's an interface. So this fungus can go out and mine 20 to 40% more nitrogen and up to 60% more phosphate, right? We're running out of phosphate mines on the planet. We've overmined it. And then it'll regulate as much nitrogen or phosphate as that plant needs at the rate that it needs it. Isn't that something, huh? And we all see the fertilizer and the Miracle-Gro and the crap, and we're going to dump it on there thinking that that's exactly what it needs. Fungi and plants have figured it out, right? If you are adding, I'll get to this in a minute. So we have pecan truffles up north. We also have pecan truffles down south. I just figured that one out. And we have these in the Carolinas on pecan trees and also oaks. And this organism here, this fungus, is responsible for 60 to 80% of our chipmunk diet in the winter. And in my territory, the upstate, it's one of the fastest growing cities and counties in the United States. And they're clearing trees at a rate where once the trees go, the biology just starts to go. You start to see animals disappear. The creeks are not shaded. Those warm up a couple degrees. What happens to the biology of the water? The trout start to recede. It all just starts there, right? So if you're looking at the biology of a soil that's been fertilized with a synthetic fertilizer, you're going to find very little to no mycorrhizae in it. You could take a little cross-section of a root, stain it on a microscope, and look at this, and you won't find any fungi in these cells. So what has happened? Now these plants are addicted to fertilizer, and they can't protect themselves. Then they can't protect themselves. What do farmers do? When it has a fungal infection, what do? They spray an antifungal compound. Now you've stripped the plant of everything. So it's like I said, if you start with the biology of mycorrhizae and knowing that all these plants have endophytes in the leaves. I'll get to that in a second. This is like one of the, I call that the axis of evil, right? This is dead biology, monoculture, right? I really like mixed cultures. When I do landscape design, I did that for years. I did edible gardens, edible landscaping around houses, mixtures of plants and different even strains. And when you're planting trees, even the trees that we have, when you're buying trees from the forestry department, what are you getting? You're also getting a tree, but you're also getting a fungus that they've selected, all right, a couple different strains. Wouldn't it be nice if the forestry department had trees that were inoculated with chanterelles and porcini and truffles? Why don't we do that, right? We should do it. It's not that hard to mycorrhize a tree. It's not that hard. Chanterelles in my area will start around June. I've got these thin little ones up here. They look like they're on steroids. And morels, like I said, just started. Absolutely wonderful. Love hunting those. And now we've got some research to suggest how to cultivate these, right? The morels are cultivatable, and you have to use the native morels that are here, right? I would never buy a culture, but I could show you how to grow those. Oh, by the way, so this guy from Ohio calls me, And he said, I was in South Carolina. He's from Ohio. He just moved to South Carolina, just like I just moved here. And then he goes, is this trout? I said, yeah. He goes, I think I found morels, but they're too big. And then he said one of the craziest things I've ever heard. He goes, can I show you where they are? And I swear, this is when Gary Linkoff used to punk me, right? And I was like, who's this, Gary? Who's this older guy? And so I said, I took the time to actually tell him. I was like, hold on a minute. You don't do that. I actually said, you really don't divulge your place, but I'll go with you. Sure, I'll go with you. Sure, you can trust me, right? So we walk up on this tree, and he said he picked 60 around one tree the day before, but he said he stopped because he didn't think he was picking morels. We picked 120 more of those around one tree. So there was 180 around one dead elm right out in the middle of a floodplain. And they were all this size, right? And you got to wear camouflage too. You got to sneak up on them. But it was insane. I'd never seen anything like it. They were all like huge, and it's on YouTube. I think it's called Killer Morel Patch, right? And you'll see the firefighter who called me. His name's Corey, right? And I protected that spot. Listen, there's fungi out there that are interesting, and I don't like to call any fungus bad. It might be it's part of the evolution, construction and destruction of the world. This is anybody know what that is?
I try to read it, but I can't.
It's a cedar apple rust.
Oh, yeah.
It's so cool. It looks like a little jelly koosh ball, right? This one is a host jumper, so it's cedar apple. It goes back and forth from a cedar and an apple tree, right? Very cool little nodules on the end of these trees, but then once those sporulate, they will attack the apple leaves and the apples, and they will disfigure those. So typically apple orchards will remove all the cedars, up to hundreds of yards, maybe a mile around the orchard to prevent this, right? How about corn smut? Anybody ever have corn smut? Anybody ever Google corn smut? Be careful. Be careful Googling corn smut, because corn might not come up first. I actually grow this, and I inject the spores right down the silk channel if you want to try to grow this. It's pretty easy, and I like eating this fresh. It's really, really good, high in lysine. It's worth more than the corn if you buy it. Another area of interest I like is the mycopesticides. This is something that the Carolinas, I'll be honest, are very rich in, but I think it just takes a slow, steady eye to find them here. When I lecture in Colorado and Telluride every year, I'd stop and pick some of the little desert sage, and I'd find the mummified grasshoppers, the little locust hoppers everywhere attached out on the grasses. So I know that these fungi are here. So once you start finding them, you can't stop. The reason I'm interested in these are because of now we have fungi that can take pesticides out of the soil, but let's not put the pesticides there in the first place. Let's try to find a target-specific fungus for these by finding them in nature. Insects, mostly insects. There are some microbial rodents. No, I can't really think of any rodents. So you have this host infection. You have this figure 8 life cycle. You see the green and the red? So the fungus exists as the green. Most of the time, it's in the environment. It's an anamorph. It's like a mold. It's eating leaves. It's eating mulch. It's just being a mold. Along comes a host insect that is target specific, right? With this fungus. It lines up, these spores stick to the cuticle of these insects. They recognize the chemistry of this particular species and they drill a hole. You've seen these before, right? And then they thread in, they get into the nerve cells They found out they get up into the brain. They thought they entered the brain, and new research showed that they don't go into the brain. They surround your brain like this. Right? Remember this right before you go to bed. And then it secretes across like the brain-blood barrier, right? Chemical activity to relocate. There's biological relocation and behavior modification. So the ant or if it's an ant, will all of a sudden start to go away. It'll never go home, right? It'll go up a tree, up a blade of grass, and it'll bite into it. The fungus melts the jaw muscles and gives it lockjaw, and then it kills the ant, and then a mushroom pops out the back of their brain. I do children's shows, and I have scared the crap out of kids, right? Maybe I'll do one at the Nature Center, because like in Oregon, this little tiny, this cute little blonde, she's sitting cross-legged on the front, and she's just like, and she goes, "Mister, can that happen to people?" And I was like, "Oh yeah." And the parents are in the back like, "What's he doing? What's he doing?" And I said, "That's what happens when you don't clean your room." And the parents were like, "Yeah." Do you have a question?
I do. Is there a target fungus for Japanese beetles that you know of?
Yeah, there is. I think I might show one. Yeah, the beetles are,
They're
problematic. Yeah.
Yeah.
Got to find them. All right. So, and then so these things. This is a biology slide. We don't need to get to that. They produce blastospores, but boom, this is in my backyard in South Carolina. This is just so common. If you want to find one of these, if you ever come down to South Carolina or Georgia in the upstate, and these persist, by the way, if you just go out and look under a branch, you will find one. If you've never found one and you're down there visiting or just hanging out, this is the easy one to find. Ant is upside down, and then it pops right out the back of his head, straight down. And they've tied strings to these, and they found out that it's not random where they position themselves, because they basically line up right with the pheromone-laden highway that the rest of the ants are using. So it's like a dirty bomb right over Highway 25, right over the overpass. Yeah, that's why I don't do kids shows anymore. I haven't been to Oregon in a while. So then you start finding these. I mean, they're everywhere. You got like stink bugs, and some of these are Todd Elliott's. He's a friend of mine. He's now getting his PhD, or he did, over in New Zealand. See, now we're getting closer and closer to some beetles, right? I found some molds on beetles. So when you find an insect, like I said, that is what I think of as agricultural, economically important. You might want to turn it in, because it's the chance of you finding that is going to be very rare, and you have to be kind of looking for it. These are aphids in a garden that got completely wiped out by this Bavaria, this little mold species. Is this economically important? Environmentally important? Of course, because people are spraying the hell out of their plants. What if we had a target-specific fungus for aphids, one for beetles, Look at this one. This one's gruesome right here. Right? This is beautiful. I love it. Listen, if you need a Halloween costume this year, look no further. Right? This one, the legs are down like the Egyptian Sphinx, right? And then the back legs are rigor mortis up in the air. And then it's got 2 mushrooms coming out the back of his brain. Right? Everybody, I want to just do a little quick a check every now. So everybody take your hand, your right or left hand, all right? Go back behind your head. All right, start at the top and just slide it down your vertebrae until oh my, did anybody feel a bump right there? Was that there when you got here? Because mine's a little bit bigger since I came here. Anyway, your homework is to measure it before the next meeting. And if you see somebody climbing the trees, just let them do it, because we need a culture. We need a fresh culture. And this is a little spider with these little, I think it's glabella growing out of it. Again, this is just right under oak leaves. So once I found the first cordyceps and I knew they were there, I was finding them everywhere. So they are here. All right, and so you'll find these, and this is a cicada nymph. You know, what are we looking at here? You know, longhorn, like Japanese beetles. Now we're looking at problematic insects. So that's why I lecture at arborist associations and tree conferences to show them these images, because I know there's a problem with beetles and other organisms, but we're not going to be the ones to find them. Arborists probably will, because they're cutting diseased wood. They're taking out trees. They're probably going to find what? A mummified beetle with fungus in it, right? They're taking the wood away and maybe burning it, but that's the opportunity, right? What about that one? Emerald ash borer, another nasty one. All right, let's find these. There should be a bounty on these. There should be a price on their little heads to find infected bugs to get them turned in. All right, scale insects. You find them. I found this one on a branch. It immediately went to mycelium, and it's a sporulating fungus on auger. All right, I think I threw out the plate twice because I thought it was contaminated. It turns out that's just what it does. All right. Bees. Paul did some work with the bees. Before Paul published his patents, he actually patented the fluid that you feed the bees. But the USDA actually did the study on Mycorrhizae that attacks the varroa mites. That was done 2 to 3 years before the bee patent. So that is a MET52. If you're taking notes, MET52, that's a Mycorrhizae. That is a strain that you could have bought and used for bees for the biological control of varroa mites. How many of you have bees? Anybody have bees? Are there any bees left? Right? Monsanto bought MET52 from Syngenta, and then they shelled it. So now you can't find MET52. Gone. This was my first cordyceps, the link of my eyelash. This was the first one I found. This is what you're looking for. So maybe about 2, maybe 4 millimeters long. And then inside the wood, I found this beautiful black carpenter ant. Absolutely gorgeous, right? My first one, I sent it to Harvard, and an expert there said, oh, we haven't seen that fungus and this ant combination since 1907 in any literature. And they said, where did you find it? And I said, I am not going to tell you. Because I was protecting the spot. But we cultured it, and it turns out this was a queen, and she had 2 mushrooms growing right out of her neck. Just like this. Everybody feel that? Look, second test. Feel that little soft spot. Right there. Come on. You're not doing it. Trust me, it'll be okay. Feel it? Now push it into it doesn't feel. Imagine 2 mushrooms coming out of there. Tonight. Right. This is probably my first and last lecture here. So I'm just gonna go for it. But we can culture this, which is amazing. You take a little piece of this and the tip, and you can make millions of that fungus with one little culture, right? And you have to be careful with this too, because this is another biological weapon, right? What else does it kill? Right? So I took this one, and then I started to play with ants. Do y'all have any biting ants here? What kind of ants do you have? Any nasty ones?
Big ones.
Big red ones. Oh, I can't wait. I can't wait to find one mummified. Right? These I had fire ants in my area. Everybody fire ants? Do you have fire ants here?
No.
Wow.
Not yet.
I can bring some if you want. So I took the black carpenter ant. This is her here. This is one of the new ones that was mummified. So I was testing the strain, the pathogenicity of the fungus. It seems like, man, it wiped it out in 3 or 4 days. So then I made fire ant farms in my house with, and then they tunneled around for like 3 to 4 days. And then after that, the fungus recognized the fire ant. So the reason I thought that might work is because a Camponotus and a Solenopsis, which are the genus of those 2 groups, are very closely related. So their chemistry might be similar, right? And this is important because I don't want it to infect other ants that are not problematic. And for me, fire ants in the southeast are extremely invasive and they cause $5 to $8 billion in damages every year, and they are whipping out the pesticides onto the lawns in bags and pallets, and it's extremely bad for the drinking water. So it's going to come back to us. So this is the way a fire ant should look, right? And I love all creatures, big and small. Love Planet, modern hippie, but these suckers. What is the alternative? The pesticides aren't the future here. Fungi is the future, right? And so this is the pesticide window you see down here. See how small it is compared even to the rest of the ants? Doesn't bother some of the native ants. The pesticides don't do that. They don't discriminate. They're gonna kill a lot of things, right? Birds, butterflies, bees, And that's the active ingredients going on the soil every year. 7.5 billion pounds of one of those ingredients goes on the U.S. soil every year. So I'm not a big fan of pesticides. All right, and this is the fire ant range here, and it's moving with our warmer climates. It's definitely encroaching. And now I hear about it in Hawaii. Now we're desperate in Hawaii and other island states. So I started studying inter-kingdom interactions. This is some work done. This is interesting. This isn't my work. I just want to point this out. But there's some plants living along the edge of these geothermal ponds here, right? And this water here is 180 degrees Fahrenheit right in here. And then it cools down to 150 right on the edge. That's the cool spot. So these plants are essentially sitting in water-drenched soil that's about 140 to 155 degrees Fahrenheit. What plant does that, right? So they found that these plants that were not colonized by the fungus were dead. And these are endophytes. So every plant out there has endophytes in the leaves and all threaded out. You don't see them. And then they purified the fungus, put it back into the plants, like a science should. They put them in the hot water and they all died again. They're like, damn, I thought we had it. We figured, I thought the fungus did it. And then a virologist looked inside of the curvularia and found a virus inside of the fungus, inside of the plant, that imparted the 155 degree tolerance. A beneficial virus, right? And so now I have a different respect for these endophytes. So in 1 year, they estimated, you might watch some old videos, that there was 1.5 million fungi on the planet. And then the next year, all of a sudden, they said, well, there may be 5.5 million. It's because of the endophyte, right? And then it's probably going to be even more than that. So as you're walking by, you may be seeing mushrooms. These are the invisible fungi that you see. And 5.5 million fungi is a lot. When we have only identified 160 to 180,000 maybe with DNA, there is a lot of work to do. We don't know anything about fungi, right? And there's fungi, if you see green plants in your garden that are doing great next to one that's doing terrible, could be an endophyte in there. Why would you save the seeds from a dying plant? Right? You're not just planting a seed, you're planting the seed. Inside the seed has the endophyte for the next generation. So if you are seed saving, now your gardening just got really fun and interesting. Because you're planting your heirloom seeds and the fungus that it protects itself. So I do workshops on how to culture endophytes. It's really interesting that when you do plant the seeds of organic seeds, you're also planting the fungal endophyte at the same time that will protect it, right? You spray that plant with an antifungal compound, you've just ruined it, right? This is kudzu. Is anybody familiar with kudzu? Do y'all have it here?
No.
Oh, so I got to bring you fire ants. And I got a whole shopping list for you. Now this is kudzu. It spreads everywhere. It just starts taking over all the native populations, just choking everything out. So, oh, I'm sorry, that's Walmart. So. And so now, the last thing I'm going to cover before the psychedelics is going to be the biologicals, which are amazing. This is staph. This is the staph that I cultured off my body. I have a certification where I can order pathogens if I want, up to level 2s or 3s, maybe level 2. Probably on the government watch list, maybe. And so, but this one, I wanted staph. I wanted a cheap staph, and so I got this one right out of my nasal cavity. Right? You can. It's all over your body, and this is normal. And if you're overusing hand sanitizers, right, just like clear cutting a forest, what's the first thing to show up on your skin? The kudzu of bacteria, right? Drug resistant stuff. So if you were washing your hands and then putting, remember all that, for how many, 2 years, with the damn sanitizer out, right? Wash your hands really good and take your hand and take your forehead right there and then wipe it back on, and it makes a film of beneficial bacteria back on your hands. It's like wearing a microbial glove. That's the real deal, right? So, sounds gross. You can also use your belly button, but I wouldn't recommend it. So this is staph, and then I started to play around. Remember those metabolites that were attacking the mold? So then I was thinking, geez, I wonder if it'll work on like bacteria, right? If it'll work on molds. Because I was looking for antifungal compounds, so I pushed in, I made some cultures, I added some E. coli that I bought from a supplier, right? Mm-hmm. So just regular E. coli, right? Get it at Walmart. You put it in there. Of course you can get it at Walmart. Just don't wash your hands. And then I put it in there, and it makes a little hydrophobic well, a perfect well. It doesn't get absorbed. It sits there, and then it starts to freak out. Right? This fungus does not like it. Fungi are territorial, remember? So inside these bags, we had controls, and boom, out sweats. Look at this metabolite, the color change, and it sweats out this fluid. And it's trying to protect itself. But at the same time, what did it do? It produced a novel antibiotic specific to E. coli, right, in this bag within 24 hours. And the other cultures didn't do it, right? One of the reasons I sold Mushroom Mountain and to become a free agent is to look back into this. I have not had time, so this is going to be a big part of my research, is novel antibiotics is going to happen, and pesticides here, right? So we have E. coli, there's the E. coli trigger, and look how many 20 milliliters came out of that one bag in 24 hours. And what's interesting is that we diluted this, and then this diluted 10 to the 6, which is 1,000,000 times, was still active, and this could produce 528 gallons of antibiotics. It was still active, and it would not allow the E. coli to grow on a plate for 30 days. So fungi know what they're doing. They produce natural disinfectants, and it's not a single molecule. If you want that, go to a pharmacy and get an antibiotic and see how fast that it becomes drug resistant. 2 years. Fungi can do this, and they're creating a cocktail to keep them from becoming drug resistant. This is, I think, the future of medicine here, right? This is interesting, right? Where we have all these fungi producing novel antibiotics, and then you have fungi here, right? I can explain this slide. I remember when my mom saw this, I had to answer some questions. She was like, Trad, what's going on there? And I said, it doesn't work. Spores are adhesive, they're sticky. And make a spore print, you can do it on glass or a mirror, and you can harvest them better, like off of glass, actually. Then they stick to paper. So I let them dry, and then I can bundle them up and make a big pile of spores for making things out of. So this is just something fun for you to do this spring. I like to think of it as, so you have, who has a bird feeder? Does the Nature Center have a bird feeder? So maybe we should make some spore prints this year of beneficial fungi. I use Hyphiloma out east, these little brick caps and things. And they're really good stump recyclers. And if we can get these stumps recycled and producing soil again and more life, so I take the spores and I mix them with the bird seed, right? And I mix it in with the bird seed, and then I fill up the bird feeders. right? And now I've created, remember, I didn't go into the Air Force. So now I've created my own branch, right, of the Earth Army. These are my F-16s coming in, right? What goes in must come out. They're out there going on bombing runs and spreading fungi all over Colorado Springs until they catch me. These are my stealth bombers, right? I think that's fun because when you look at a destructive habitat, it can take 600 years to make 1 inch of soil, right? And fungi can do it way quicker with our help. They need our help now. They've helped us get this far, and now we need to help fungi make soil quickly, right? This is our future as well. We've got to partner up with fungi to create and make soil here. There's also polyurethane degrading fungi. This is Psilocybe. You can read about this. Yale did a lot of these discoveries. Another endophyte. This wasn't a mushroom they found. They punched a leaf in the Amazon, and out comes a fungus out of a leaf. It could potentially be used to eat plastic. It's wild, but this is the forest that I want to wander around in. Not the clear cut, not next to the creek with the dead trout. Right? Fungi are interface dwellers, and we really need to get them where they need to go. Right? That's our future. And speaking of our future, right, is the psilocybin and psychedelics. So the Blue Portal is a company I started, and I met Irene, was it 3 years ago? About 3.5, now 4 years ago. Before COVID was shut down my lab in Jamaica, I was working in a lab in Jamaica, and I was growing for a Canadian company and myself, and we were doing retreats there. All right, so I've been growing a lot of these strains down there, and now, whoa, I want to just see who's, no reaction yet. Okay, everybody's clean. There's something taped to your chairs. $2 grand. So anyway, what I want to tell you is that the world really does, the work that we do has been highly effective. I could never go back to what I was doing after running the Blue Portal, because I've seen not just a need for it, and I hate to use the word like a business. I don't even feel like I run the business, but we do. But the transformation that's occurring with participants it's unbelievable. And when I talk about it, I get goosebumps and I choke up, because I've seen just how wonderful humanity is and can be, And this really should be mandatory, right? I hate to say it. It should be in the drinking water, guys. But don't do it. Don't do that. So unconditional love. Like, it's okay to hug a chicken. This is Chicken of the Woods, by the way, if you've never seen it. I got some cheesy jokes, man. I don't know. We doing it this long? But one of the things about fungi that has taught me, and I did spend some time in the Middle East when I was young, in Egypt, and it was a interesting year for me. But what I see is this out there. Mushrooms can't talk our language. They don't speak like human language, but they speak other languages, and we just have to pay attention to what they're telling us. Like the Petri plate. I see that he's attacking this one. He's telling me, look at what I can do. What are you going to do with this, huh? We got to watch them, and we may be able to take notes and learn a little bit of fungal empathy here, because, we've not been on this planet a very long time. If we want to, let's take notes from something that has, right? A very long time. So there's the Rosetta Stone. Oh, and there's Irene Dubin right there. Do you want to say anything about what we do? Come on, Irene.
You can cover that.
All right, so we've got the Blue Portal was something I started in, again, in Jamaica, and it was a part of Mushroom Mountain, but I took it, and there's a lot of work being done on the psychedelic front, all right, and most of our work is being done in Jamaica. I cannot do it in South Carolina, at all? Anyone from the southeast? If you were caught cultivating a certain number, you would get 20 years in prison, mandatory sentence. That is more jail time than robbing a bank. So I figure I just rob a bank, right? But 20 years in prison for that is just ridiculous. I'm happy to see that Colorado is moving towards, I know we're just keeping an eye on it, yeah. And Oregon, I don't know about what's going on in Oregon yet. But we do keep up with the industry and trying to see what's going on. I'm also very interested in what's going on with the medical model versus the ceremony and community model, right? And so I will tell you that 2 people that moved here are part of the community model, not the medical model. So we're here to live and look for mushrooms and things, but we're keeping our business abroad at the minute, just for legality and safety reasons. So trying to think if there's anything else I could tell you. And also Irene is really good with that. She lectured at Telluride and did the Amanita muscaria talk, which was really good. Did you see it?
I have notes on me.
Yeah, notes on you. So we have so much to offer, but I really I hope you do too. This is a whole new area for us in a region, so hope this is what I do. I don't have cable, so I cut out shapes and watch them grow. Like I said, and I really am hopeful that I'm really hopeful and glad to be here. And now that we're here in this little mountain village with open minds, that I can write another book and really start to dig into the antibiotics and more about what mushrooms can do. And I just couldn't get that feeling from where I came from. So I'm breaking out and coming here. So love to see all the smiling faces. So peace, if we have peace, right, you can have love. You might be able to buy love, but damn it, you can grow it. Right? Let's say. What's cool is you cut this out and it just grows. You get to watch it grow. Right? And there's peace and love and, of course, happiness. Right? And mushrooms, I said, it's the greatest thing. I'm really at 18, 19 years old to be here still studying mushrooms is just absolutely amazing. So I know you are an amazing group, and we've got a lot of cool stuff ahead of all of us. And thank you for coming tonight. And we're going to hang out and answer some questions if you like. So thank you so much.